this basically model, we have three main sections. I don't know if you have seen it, A and B and C, because the skin is different in different parts of our body. For example, it's easy to tell that the skin in the palm of our hands is thicker than the one in our eyelids or things like that. And so you have thicker areas with smaller areas of the epidermis. The epidermis is this portion here. In this thick area is from here to here. The dermis is from here to here, okay, and the hypodermis is from here to here. Okay. In this other portion, the epidermis will be from here to here. The dermis will be from there to here, and the hypodermis, all this portion. Same thing on this area. In the epidermis, the type of epithelial tissue, okay, is going to be stratified, squamous, keratinized, or cornified. It contains 5 layers. It contains the cornified layers, the lucidum layers, this white portion there, okay, the granulosum layer, this dark area, okay, the spinosum layer, all this area that looks lighter in color, and it goes deep in there. All that area is going to be the spinosum layer, and then the basal layer, which in this case is represented only by a single layer of darker cells, okay, and it goes up and down, up and down, up and down. That is the 5 layers of the epidermis. The lucidum layer, which is this white portion, only is present in thick areas of the skin, like the palm or the sole of the feet, the palm of the hand. You don't see that in the other areas. You have the corneum layers here, the granulosum, the spinosum, and the basal, but no lucidum layer. All right. The dermis contains 2 main regular layers. The papillary layer, which go from here all the way from this area all the way to this area in there. So all those will be papillary layers, okay? And then the reticular layer, which go from there to here. The papillary layer consists of the areolar connective tissue, and the reticular layer is the dense irregular connective tissue. What is the function of the dense irregular connective tissue? Is to allow movements in multiple directions. That's why it's called irregular. What is the function of the areolar connective tissue? The function of the areolar connective tissue is to allow the play in between the epidermis and the dermis. These shearing forces that I'm doing in my skin here, okay, doesn't cause blister. Why is that? Because of the areolar connective tissue there on the one hand, and it doesn't cause bruises because of the irregularity of the collagen fibers in the dense irregular connective tissue. All right. What structure do we find in the papillary layer? Well, on the one hand, we have these little funny structures, oval structure. That is the Meissner corpuscle. Okay, and that is the function of the Meissner corpuscle is a light touch, superficial touch. Okay. We also find plexuses, where plexus means just a network, in this case of blood vessels. The red ones will be arterioles, and the blue ones will be venules. Okay? And let me think here. Okay, that's all we can see there. In the reticular layer of the dermis, we find several structures. We have found glands. Okay? We have several kinds of glands. We also have hair follicles. This will be skin that is mostly in the scalp. This is a skin that is mostly in the armpits. This is a skin that is mostly in the sole of the feet or the palms of the hand. Notice that in the sole of the feet you have mostly this kind of sweat glands. These white portions are sweat glands. This is a merocrine sweat gland, merocrine sweat gland. In the armpit, we also have merocrine sweat glands, but we have these that are called apocrine sweat glands. Both are sweat glands, but this is merocrine and this is apocrine. The mode of secretion of both is the same, merocrine. The type of secretion is a little bit different. This is more watery. This is a little bit more thicker. You find them mostly in the armpits, which is actually called axillary region. You also find in the anogenital area. Okay. In addition to that, you have hair follicles. The hair follicles contains sebaceous glands. This is a sebaceous gland. You also have another sebaceous gland here. These are sebaceous glands, obviously producing sebum, not sweat. The product of the apocrine sweat gland is sweat. The product of the merocrine sweat gland is sweat. The product of the sebaceous gland is sebum, S-E-B-U-M, sebum, not oil, sebum. Okay? The mode of secretion of the sebaceous gland is holocrine. I told you that the mode of secretion of the merocrine sweat gland is merocrine. The mode of secretion of the apocrine gland is also merocrine, but the mode of secretion of the sebaceous gland is holocrine. What's the difference? The merocrine secretion is the cell secretes by exocytosis. That is, the Golgi apparatus, which you all know already, those of you who have taken biology 1, the Golgi apparatus produce vesicles. The vesicles move to the surface of the cell, and then by exocytosis release the secretion to the outside. What about the sebaceous gland? The sebaceous gland, the cell rupture is full of vesicles. The Golgi apparatus produces the vesicles, is full of vesicles, and is packed, and then it bursts open, and it releases all those vesicles with the sebum to the outside. So that means the cells die. So that means here in the darker area is stem cells that have to be replaced constantly every time that this gland produces a sebum. All right. Notice that this case, this sebaceous gland is attached to the hair. Not all the sebaceous glands are attached to the hair, but in this model, that's the only one that we have here. The hair follicle. The hair follicle consists of the shaft, the glandular area. This is the shaft from here up. The glandular area, which is this portion there. This portion here, where this muscle attached, okay, this portion is actually called bulge, like a bulging, and then this portion here, which is called bulb, B-U-L-B, bulge, B-U-L-G-E, bulge. In the bulge area, the cells that are here are the stem cells that will grow and actually makes here the papilla, what it is, matrical cells. This is the hair matrix. Matrical cells, hair matrix, okay, to continue maintaining the growth of the hair constantly. Okay. How do we distinguish where is the bulge versus the bulb? The bulge is always where the muscle attached. What kind of a muscle is this? This is a smooth muscle. The name of this muscle is arrector pili muscle, not erector, is rather arrector, that is with an A, letter A, arrector pili muscle. It's a smooth muscle, and the function is to cause the hair to stick up, in other words, to give you the goosebumps. So therefore, it's very important in terms of regulation activity. You have a bunch, obviously, in the scalp. All right. Then from here down here, okay, is the hypodermis. So from here to here is the cutaneous membrane. From here to here is the subcutaneous layer or hypodermis. It consists of two main types of connective tissue. These yellow portions is the adipose tissue, which is part of the connective tissue, and these areas here is the areolar connective tissue. This areolar connective tissue allows play in between this package of fat cells and this package of fat cells. The fat cell is actually called adipocytes. Okay? In this model, we have this other structure embedded in the adipose tissue. This is the Pacinian corpuscle. The main function for this is a deep touch. Okay, deep touch. The Meissner was superficial touch or light touch. The Pacinian is deep touch, deep touch. And I believe we have a microscope slide of both, so it's better that you go and try to identify it in the microscope slides. And so both of these are neural tissue. Both of those are neural tissue. The Pacinian is not only found in the hypodermis, we can also find it in the deep layers of the reticular layer of the dermis, in the deep portions of the reticular layer of the dermis. We also have a plexus here, which is actually the subcutaneous plexus. I forgot to mention this is the papillary plexus. This is the subcutaneous plexus. When you do an subcutaneous injection, you have to introduce the needle all the way to the subcutaneous area. So what kind of questions I tend to throw in the practical exams? I might put a sticker here, a label. They say label number 3, right? And then I might ask you, name the type of tissue located at the label number 3. And then you look at where the label number 3 is located. So what would be the answer?
Areola.
Areolar? No.
Dense.
Dense irregular connective tissue. Very good. What if I put them in one of these bumps?
Areolar.
Areolar, because these are the papillary layers. What about if I put them anywhere here?
Epithelial.
Stratified squamous keratinized.
Very good. Stratified squamous keratinized epithelium. Another type of question that I tend to ask is, I put a label here and then I said name the layer. Spinosum, stratum spinosum or spinosum layer. Either one of the two will be correct. Okay, and there is also a microscope slide that I can point at what layer is that. So you need to know it in the model and also in the microscope slide.
Is the whole thing keratinized or just the corner?